Hello, this is Alice Shapiro for HO 152, Human Body Systems 2. This is the second part of the recording for the section on lymphatic system immunology and microbiology. So we had left off with talking about adaptive immunity, and we are now into the section that is talking about antibody-mediated immunity, and we're now talking about antigen recognition. Lymphocytes have antigen receptors. Remember, an antigen is a foreign substance in the body. They are called B cell receptors on the B cells and T cell receptors on the T cells. And each receptor binds only with a specific antigen. So they are very specific. Sometimes adaptive immunity is also called specific immunity. When antigen receptors combine with antigens, the lymphocyte is activated and the adaptive immunity begins. So this specificity begins. There is a complex of histocompatibility molecules that contain the actual binding sites, and they are specific for certain antigens. They hold and present a processed antigen on the surface of the cell membrane, and then they bind to the antigen receptor on B or T cells and stimulate the response. So these are cells that aid and assist in this process. Cytokines are other chemicals involved. They are proteins secreted by a cell that regulate the neighboring cells. For example, one type of cytokine is interleukin-1. It is released by macrophages, and it stimulates helper T cells to recognize the foreign invader. Here is a summary that shows you the proliferation of helper T cells. Again, this is a good review. I'm not going to talk about it all now. This is available in your textbook, so please go back and look at it either on the non-narrated portion or on your textbook. It provides an excellent summary. Lymphocytes then proliferate after an antigen is processed. The helper T cell produces various interleukins and interleukin receptors, and the interleukin binds to the receptors and stimulates the production of more T cells. And the helper T cells are needed to produce the B cells, and the B cells produce the antibodies. So there are a number of cells involved. Here is a summary. Please, again, take a look at this. This is in your textbook. It's also available non-narrated on your Moodle site. There is a dual nature of the immune system. Actually, I would say even more than a dual nature. There's a dual nature of the adaptive immune system, both the antibody-mediated and the cell-mediated immune responses. Antigens can trigger both types of responses, and both types are able to recognize self versus non-self. Both have specificity, and both have memory. The antibody-mediated immunity is effective against antigens in the body fluid, so in the blood and lymph. It's most effective against bacteria, viruses, and other toxins, and the B cells are involved. The B cells produce antibodies. Antibody structure is basically the shape of the letter Y, and you don't have to memorize the different regions, but they bind to an epitope, which are the recognition sites, we could say, on the antigen. So take a look at this just to familiarize yourself. Again, this diagram is available in the non-narrated portion and also in your textbook. You don't have to memorize this, but get familiar with what is going on here. The antigen binding site is the site on the antibody where the antigen binds. So these are some definitions. Valence is the number of antigen binding sites on the antibody, and there are 5 immunoglobulins used to destroy antigens, and they are listed here for you. I'm not going to ask you to memorize all of these types, but be aware that there are 5 types. Antibodies inactivate the antigen. They bind them together, so they agglutinate them. They activate complement molecules, which help to destroy them. They initiate the response, the inflammatory response, and they facilitate phagocytosis. So here is a summary of the actions of antibodies illustrated for you. Again, this is in your textbook and in the non-narrated portion. Please take a look at this as a summary for yourselves. Antibody production. The primary response is the first exposure of a B cell to an antigen. The B cell undergoes division and forms plasma cells and memory cells, and it is the plasma cells that actually produce the antibodies. And at the first exposure, it takes 3 to 14 days to be effective against the antigen. So the person will develop disease symptoms. The secondary response involves memory cells. So once you have been exposed to something, your B cells produce B memory cells, and they quickly divide to form plasma cells, much more quickly, which produce antibodies and produce new memory cells. So the second time you are exposed, your body is much more effective in fighting off the invader. And here is an illustration of the primary response, which is slower, versus the secondary response, which involves memory. So you can see this time charted here for you. Cell-mediated immunity is effective against antigens in cells and tissues. It is effective against bacteria, viruses, fungi, and protozoa. And this uses the T cells, which remember matured in the thymus gland. There are several types of T cells, and I'm not, again, going to ask you to memorize all of these, but you can see this is a very complex process. So take a look at these again later on. And here is a summary of T cell actions. This is very helpful to help you to remember and put this all kind of together in perspective. And this is a summary, again, of what the T cells will do and how they interact with one another and what other cascades of chemicals they produce. And this kind of gives you a division between antibody-mediated immunity and cell-mediated immunity. Notice all of these types of immunity, whether it is innate or adaptive, cell-mediated or antibody-mediated, do work together. We don't live in an isolated system where one part of our immune system says, oh no, I can't do this, I'm supposed to be adaptive. They all work together. We divide them so we can understand them a little bit better. All right, types of adaptive immunity can be active, which involves natural exposure to the antigens and produces the production of antibodies. Or passive, we can transfer antibodies from mother to child, such as in breast milk or from the placenta. Or artificially acquired immunity, active would be an injection of antigens, and this process introduces killed live or inactivated or attenuated pathogens. Or passive is the injection of antibodies from another person or animal. And here is a summary of those processes. Now, just briefly go over some microbiology. First, looking at bacteria, and again, this is from your packet and from the published material on our Moodle site. This is not in your textbook, but you have plenty of time on the non-narrated portion to take another look at this. So we can have coccus, bacillus, which are rod-shaped, or corkscrew-shaped, which are spirillum. Molds also cause disease. This is histoplasmosis, and this is an example of a fungi that causes disease, Candida albicans, and that's a pathogenic fungus, which is a major health problem. It is often suppressed because it is in competition with other organisms, but it can become a problem when a person is treated with antibiotics or when the immunity drops, such as in HIV infection, which leads to AIDS, or drugs that are used to suppress an organ transplant reaction. This is a type of pathogenic cell, an amoeba, a one-celled organism. And this is Giardia. This is a diagram of a cilia to show you the cilia which are involved in helping this microorganism to move. And these are examples of paramecium. And there are some websites where you can do a little further investigation. And also, sporozoans are parasites that, for example, can cause malaria. And this shows you how malaria can be conveyed. This sporozoan is transmitted to people via the mosquito. And here is a life cycle of a typical virus. Again, this diagram is available on your Moodle page, but let me go over this. This may not be as clear. This shows you a virus particle attaching to a host cell. It is ingested via endocytosis. Viruses cannot produce independently. They must have a host cell. When they invade a host cell, their RNA, usually it's RNA, sometimes it's DNA, will take over, will generally embed itself in the DNA of the host, will cause the host cell to stop its own processes and simply be involved in producing more virus particles. And here are the virus particles being produced. When enough virus particles are produced so that the cell is full of virus particles, the cell will burst and die, and as many as from one viral invader, as many as 100, we could say offspring virus cells will be produced and will be then released and can infect, each one can infect another host cell. So you can see the geometric speed at which these viral particles can evolve, can reproduce, I should say, multiply. Here's HIV replication life cycle, similar to what I showed you. HIV is a virus, and it does invade the cell, and it enters the nucleus of the host cell and takes over the processes. New virus particles are produced, they leave the nucleus, and they assemble at the cell surface and then are released, and often the host cell does burst. All right, make sure you review the packet that goes along with this unit and go back to the non-narrated portion. If you have any questions, please do notify me. And this is actually the final recording, narrated recording of the PowerPoints for this term. So I will be in touch with you again. Messaging me via Moodle is the quickest way, and I wish you happy studying and good results. All right, that's all for now.